In this topic, we're going to discuss the uptake of water and the transport across roots. So I'm going to outline the transport of water in a plant. Then we're going to look at root anatomy, the uptake of water by root hair cells, the transport of water across the cortex via the apoplast, symplast, and vacuolar pathways. And then I'm also going to discuss root pressure. So water gets transported through the different parts of the plant. It starts off with the uptake of water into the root hair cells via osmosis. Then the water moves across the root cortex to the xylem. It moves up the xylem. It moves from the xylem into the leaf cells. And then it evaporates from the mesophyll cells into the air spaces. And finally, the water vapor moves out of the stomata, and we call this transpiration. Looking at the structure of a root, if you look at a root under a microscope, you'll notice the root tip is covered in a hard, protective, non-permeable cap. So this is called the root cap, and it's mostly dead cells. Then just behind this is the meristematic tissue. This is where cell division takes place. Then you can see the region of cell elongation. And finally, the region of maturation and specialization. So once the cells specialize, some epidermal cells will form root hairs. So these are cytoplasmic extensions between the soil particles. And this is where water enters the root. So they provide a large surface area for water absorption. They are quite delicate, so they only last for a few days and need to be replaced. Now, some plants have a symbiotic relationship with fungi called mycorrhizae. So these increase the surface area for water uptake, and in return they gain organic nutrients from the plant. So they behave like root hairs. Okay, moving on to root anatomy. Just to recap, the xylem, which carries water and mineral salts, is found in a star shape in the center of the root. So here you can see its walls stain red in the picture of in the left there. Now notice the other parts of the root are the outer cortex, epidermal root hairs. Remember that the xylem and phloem form the stele. So take a moment to look at this picture of the root anatomy. Can you remember the different parts? So we've covered this topic in the topic distribution of vascular tissues in plants. So the epidermis contains root hairs in young roots, and it gets replaced by a protective exodermis. The cortex is made of parenchyma, and this may be for storage. You've got the xylem and phloem in the center there that form the stele, and then this is surrounded by the endodermis and pericycle. Remember that the pericycle is meristematic and the endodermis has got a layer of suberin. So now that we've looked at the structure of the root, let's have a look at how water is absorbed into the root. So surrounding the root hair cells, you've got soil and soil water. And the soil water has some inorganic salts, but it is relatively dilute. This means that it has a higher water potential, only slightly less than 0. Inside the roots, there are sugars, amino acids, and mineral ions dissolved. This means that the root hair cells, or the root cells, have a lower water potential. So water moves from a high water potential from the soil water into the root hair cells. down its water potential gradient by osmosis. So it moves through the partially permeable membrane of the root hair cell. Remember that all water potential values of solutions are negative. So water moves from a higher, which is a less negative value, to a lower water potential, which is a more negative value. Now that the water has moved from the root hair cell, where is it going to go? It's going to cross the root cortex to the xylem. So let's have a look at the different pathways. So once the water is absorbed through the root hair cell, it continues its journey across the root cortex in different ways. So you've got the apoplast pathway, the symplast and vacuolar pathway. The apoplast pathway involves the movement through the free spaces between the cellulose cell walls, and this forms a continuous stream of water. The symplast pathway takes place through the cytoplasm of the cells of the cortex, whilst the vacuolar pathway involves water moving through the cytoplasm as well as the vacuoles. Looking at the apoplast pathway in more detail, so what happens is it pulls more water along with it due to the cohesive properties of the water molecules. This creates a tension and draws more water along the cell walls of the cells of the root cortex. So the mesh-like structure of the cellulose cell wall of the cortical cells has got many water-filled spaces. So there's little or no resistance to this pull of water along the cell walls. The symplast and vacuolar pathways take place across the cytoplasm and vacuoles of the cortical cells as a result of osmosis. So the water passes through the cells along tiny openings called plasmodesmata. Each plasmodesma is filled with a thin strand of cytoplasm. There's therefore a continuous column of cytoplasm extending from the root hair cell to the xylem at the center of the root. So as water moves from one cell to the other, a water potential gradient is set up. So this is a lovely diagram to show you how the water moves via the different pathways. You've got the symplast pathway and the apoplast and vacuolar pathways. Now remember that the cells in the stele are surrounded by a band of waxy waterproof suberin, which forms the Casparian strip. This blocks the apoplast pathway and forces the water to move into the symplast pathway. So here you can see the waxy Casparian strip of endodermal cells blocking the apoplast transfer water from the cortex to the stele. Notice how the water is forced to move through the symplast pathway. So this allows for the control of water, mineral salts, and toxins entering the xylem. Now in older roots, most endodermal cells are fully suberized, and water can only move through a few non-suberized passage cells. Active transport of salts is the most likely mechanism by which water now gets into the xylem. So by pumping nitrates into the xylem, a much lower water potential is created, and this causes water to enter the xylem from the endodermal cells by osmosis. As this active process requires energy. It can only occur within living tissue, so it therefore takes place along carrier proteins in the cell membrane. So this explains why the water from the apoplast pathway is forced into the living protoplast of the cell by the suberin band of the endodermal cells. Now there's something called root pressure. So the active transport of mineral salts into the xylem creates a lower water potential in the xylem, and the subsequent osmotic movement of water into it. This creates a force that helps to move water up the plant, and this force is called the root pressure. So it's what causes water to leak from newly cut stems. It contributes to the movement of water up small plants, but it is inadequate to account for the movement of water up large trees. So evidence for root pressure. The pressure increases with a rise in temperature and decreases at lower temperatures. Metabolic inhibitors, for example cyanide, prevent most ATP production by respiration and therefore cause root pressure to cease. And finally, a decrease in the availability of oxygen or respiratory substrates causes a reduction in root pressure. So in summary, what have we looked at in this topic? We outlined the transport of water in a plant. We looked at root anatomy. We discussed the uptake of water by root hair cells, the transport of water across the cortex via the apoplast, symplast, and vacuolar pathways. And then we also discussed root pressure. And that concludes our lesson. The end.